everyone. Welcome to the 17th talk of capstalks series. So last week we heard about the omics world of medicinal plants part 1, and today we will be going deep into the part 2. Over to you, Vikas.
Thank you, Shailya, and good afternoon, everyone. So we are continuing this theme of omics world of medicinal plants, and this is the second part of that. And today we will be talking about two plants, two species of Moringa plant and one for the Sankapushpi plant. So the drumstick plant or the Moringa plant is also known as the magic tree because of its several medicinal properties, which includes anti-diabetic, anti-obesity, and also it protects from the it has cardioprotective properties as well. And different tissues of the drumstick tree has these medicinal properties. So the goal or the lab was interested to understand the transcriptome of these different tissues from this plant, and with the emphasis on the expression of genes involved in the synthesis of these several secondary metabolites which impart these medicinal properties to this plant. So we wanted to quantify the amount of these secondary metabolites, the vitamins and or the transporters, genes, gene expression levels from these different tissues. And entire lab was involved in this transcriptome analysis of this drumstick plant, and especially Dr. Nasir, who is with us, and he will be talking in detail about it. And he has this innate ability to go after a problem, and he had his way way with people to solve the to come up with the solution for these problems. And being a computational lab for performing several wet lab experiments, for example, doing some quantification of minerals, he could just walk into the other wet lab person's lab, and he'll get the job done. So with this brief introduction, I would invite Dr. Nasir to explain this work in detail. Over to you, Dr. Nasir.
Yeah, thank you very much, Vikas, for that introduction. So firstly, I want to thank ma'am and the lab for giving me this opportunity. I think this initiative on plant genomics, focusing on Indian medicinal plants, originated around 2013-14. And I'm glad that I was part of that journey. And today what I'll be talking is one of the medicinal plant called Moringa oleifera. And I would be briefly talking about it in a touch base about this plant and the kind of work we have done. But more importantly, I will be talking about the vision that ma'am had on this plant genomics initiative, where the main objective was to identify what are the key metabolic enzymes underlying in the biosynthesis of secondary metabolites that has medicinal values. So, and Moringa oleifera was one of the shortlisted tree. And the reason we wanted to focus on this is Moringa, as even Vikas mentioned in his introduction, it's called as the magic tree or the miracle tree. But it belongs to, just a quick introduction on the botany side of it, it belongs to a genus Moringa, species Moringa oleifera. And there are already around 13 species of Moringa oleifera, of Moringa genus, and Moringa oleifera and Moringa concanensis are the Indian origin species. And our first effort was on Moringa oleifera to understand their medicinal values using transcriptomic analysis. And as you can see the name Miracle Tree, then there must be a reason for it. And there was few quantifications done previously by a few scientists, as you can see on the table listed below. So these are the vitamins and the microelements, and was compared across Moringa and the other foods that are part of our diet. And all of those, if you see those numbers, they're almost like 5-folds or 10-folds more than what you see in your regular diet, which it's suggesting that the importance of this particular tree. So coming to this study, what we did in the lab was to do a comparative analysis of 5 tissues of Moringa oleifera. So we used flower, leaf, seed, root, and stem, generated RNA-seq reads. We isolated RNA from each of these tissues, used Illumina platform, got the raw reads for each of this. Now, the first question comes is to quantify the genes, the abundance of the transcripts. So for this, at the same time, there was a Chinese reference, Moringa reference genome was released. So we used a reference-based transcriptomic assembly and mapped each of these reads to the reference genomes and quantify the transcripts. So, and then following quantifying the transcripts, we did a differential gene expression analysis. So that to know what of the transcripts are highly upregulated or downregulated across these different tissues. Once we had the data of expression data across these tissues, the next question we asked was, now there are a list of secondary metabolites that has medicinal values in this plant. The enzymes involved in those secondary metabolites, which part of the tissue is highly enriched versus not. So for that, there was a in-house pipeline developed across it, which I'll be coming to it in a couple of minutes, and identified those transcripts that are highly abundant, that are involved in the secondary metabolite biosynthesis. And similar thing was done for the vitamins and the transporters. And then these genes were validated using RT-PCR analysis. And in few cases like quercetin, we also used HPLC to quantify the amount across these tissues. So this was from a transcriptomic perspective. But as I already mentioned, there was already a Chinese genome that was released more or less at the same time when we were working on it, and had a higher genome coverage because of they had integrated long reads into it. So we also had assembled a de novo assembly for the Moringa. And since this is a transcriptomic assembly, we were able to have a size of 79.79 MB. But interestingly, if we only focus on the gene models, we were almost like around 17,148 genes had, we were able to predict those gene models in comparison to Chinese genome assembly, which was around 19,465. Following that comparison, we also did orthology comparison across different species, and the list have been shown here. What we saw here was in the Venn diagram, what you see are the gene clusters that are, you can see the overlap of genes and also the unique set of gene clusters, as well as what you see on phylogeny is we saw based on the phylogeny using these orthologous clusters, we saw it was much closer to Carica papaya, which belongs to Caricaceae. So in terms of the completion of gene models, we use a tool called POSCO, where around 83.8% of genes what we predicted were of complete, and 4.4% were fragmented, and 11.8% were missing. So, and these numbers are pretty much at those times were impressive. And following, as I was mentioning about, the main objective was to identify the enzymes and their expression across these tissues. There was a pipeline that was developed in-house. Now, assume, let's say we have a list of secondary metabolites, and from different sources, we know their medicinal values and so on, and identify what enzymes are involved in those metabolic biosynthesis, and see whether those enzymes are observed in our transcriptomic data. So to do that, you take those from literatures and other public resources, those enzymes, and do a homologous search in the Moringa transcriptome data that we generated. And then following, we would combine with the ortholog sequences and along with the sequences that we generated during the study, and identify the functionally important residues. And you eliminate those sequences, those transcripts where you don't see those conservations in terms of the functionally important residues, and only retain a use for further analysis where you see those FIR regions. Further, we would validate these sequences using qRT-PCRs and so on. So this was overall, I know Advait from previous presentation, he has discussed in more detail. So I would not go much deeper, but if we take an example on this, if you take an example of quercetin, which has a lot of medicinal values in terms of anti-cancer and so on, there are 6 steps involved in it, starting with phenylalanine as a starting substrate and ends with the quercetin. If you take an example of the 3rd step, where there is an enzyme chalcone flavone isomerase involvement. So now we would use this sequence and identify the ortholog sequences from a well annotated database like UniProt, and do a multiple sequence alignment with those ortholog sequences and combine with the transcripts that we generated in our analysis. And for instance, if you see, we had 2 transcripts. Now the question comes, which of these transcripts have to be would be part of the actual mechanism in the biosynthesis. So and then we would generate a phylogeny from this, and we saw that one of the transcripts were co-clustering with the annotated sequences, and then we shortlist them and validate using some wet lab experiments like qRT-PCR. So same approach was used for multiple secondary metabolites like camphorol, quercetin, benzylamine, and so on. And not only in terms of secondary metabolites, we also focused on transporters and vitamins. What you see on my right side was the secondary metabolites that we focused, and left-hand side are the enzymes that are involved in those secondary metabolites. And the heat map depicts that the expression profile across these different tissues. And as I mentioned this, if all of these transcripts were again validated using wet lab experiments to add more value to our data. And for especially for one of our secondary metabolites focus, that was quercetin, we also did a HPLC quantification, and we observed that there was a high amount of quercetin in flower, following leaf, and so on. So that was with respect to the transcriptomic analysis and comparing secondary metabolites across different tissues and vitamins. But we also did another interesting analysis, as you would come across in a lot of literatures, and that the Moringa leafera has higher amount of microelements. What I mean by that is like aluminum, copper, calcium, and so on. All of these microelements are very much important, and some of these elements also act as a cofactor in few enzymes. So what we did was, we wanted to, since we are doing a comparative analysis across different tissues, we wanted to quantify these microelements in each of these tissues and understand which of this tissue has higher elements. So for this, we used a technique called as inductively coupled plasma atomic emission spectrometry. And this was in collaboration with Dr. Mahesh Shankaran's lab. And they use it for some wildlife conservation purpose. But I think we thought that we could leverage the same technology and quantify these elements. So for this, we had used spinach as a control, and what you see in the 3rd column are those values for each of these elements. And following what you see are the different 5 tissues that were used in these tissues. As you can see, there were high amount of elements, especially in the roots, in comparison to other. As an exception, like potassium was observed in throughout all of these tissues were in higher quantity. And it pretty much reflected what we saw in the literatures in the previous studies as well. So this figure is one of my favorite, which Shafi had really helped us in putting together with MAMS and other teammates support. Basically, what it tells is the comparative analysis of these 5 tissues and the secondary metabolite, the enzymes involved in the secondary metabolites, which part of these tissues are highly enriched, and not only in terms of secondary metabolite, even the vitamins and the microelements that we were talking about. So in a way, it kind of sums up what we have done in this particular study. But I just want to now shift your attention to a little different, because the objective was not to talk too much science behind the work that we have done here, because it's pretty much captured in the literature. But what we want to say, how relevant is this kind of studies in today's scenarios? So if you see, if the kind of there's one kind of sentence that I had mentioned here, because there's from a traditional medicine, that is from Ayurvedic perspective. In Ayurveda, especially Moringa, the roots and barks are used for medical purposes and to pacify something called as kapha. So people who are not aware of all of the term called kapha is in Ayurveda. In Ayurveda, there are fundamental elements called vata, pitta, kapha, which are kind of represents energies from uh, wind, fire, and water. And every individual needs to have a certain kind of balance of all of these elements. So this so if you see kind of from a conceptual point of it, basically traditional medicine is trying to stratify patients and saying that each of these patients require specific kind of a treatment. Now if we come back to the modern science, that's what exactly we have been doing. Nowadays we talk that we have to use multi-omics approaches, use combining DNA, transcriptome, proteome, metabolome to stratify the patients and understand the pharmacogenomics of each of these stratified patients, and they and give an understand in terms of who would respond versus who would not respond to the particular drug. So conceptually, you can see that there is some level of similarity. The point I wanted to make is that the concept that we have been discussing today in the modern science, that's been already being applied uh, in traditional medicine like Ayurveda. So, and this kind of a study, and the reason there is a gap between the traditional medicine and the modern medicine is because of the flow of information from both the medicines. So I think this kind of study would really help the to close the gap between this traditional medicine versus the modern medicine. So yeah, so this work was also featured in very high profile medium like Deccan Herald and I think the Hindu. And I think this was pretty much also important that we could convey our research into very layman terms so that they can also understand what's going in terms of research. And it's I personally feel it's pretty much it's important because they will also be aware and have a sense that what we are doing with their money. Yeah, finally, I just want to thank ma'am, and what you see here in this, because only few representative, but the team was huge. It was a collective effort, and we need thanks to NCBS and CCAMP for the facilities and infrastructure, and we had a DBT funding for this work. So I think I'll stop here and be happy to take questions.
Thank you, Nasir, for the wonderful talk. Okay, so that was an excellent talk by Nasir. Moving on, we will be hearing about another species from the Moringa itself, which was mentioned by the Nasir, which is Moringa concanensis. It is found in the Konkan region of India, and like Moringa oleifera, it is also used traditionally for several medicinal purposes, but very little scientific information is known about this particular species. And therefore, the focus was to do the comparative study between the Moringa oleifera and the Moringa concanensis, with focus on the secondary metabolites which are implicated in the anti-diabetic activities. So it was done again from transcriptome perspective, and it will be explained in detail by Dr. Shafir. Over to you, Shafir.
Thank you, Vikas. So, hello everyone. So, Nasir nicely talked about the common transcriptome. So, I'll be talking about another species which is mostly found in India, and we have compared this species to the previous drumstick tree. So, in Moringaceae family, there are 13 different species. and most of them are found in African countries. And what you can see here, the first Moringa olifera, Moringa congrensis, even Moringa peregrina, these all you can find in Asian, and first two are mostly found in India only. They are recorded from India. So as Nasir told, Moringa olifera, that tree is highly drought tolerant and it's a fast growing tree, and this involved in many medicinal or nutritive properties. Hence, this plant is cultivating worldwide currently. And for Moringa congrensis, it's a small tree. It highly resembles to Moringa olifera, and is mostly found in Konkan region in India. And there is not much scientific information known so far, but it's traditionally used as a medicinal plant. Now, I told it highly resembles to Moringa olifera, but we can see clear differences between these two species. Like this Moringa olifera leaf, it's highly dense, but where you see the Moringa congrensis leaf is highly, it's big and it's oblong. And if you look at the flower, it's cream white flower for Moringa olifera, but a yellowish petal and pink vein you can see in Moringa congrensis. And if you look at the therapeutic potential, there are a large number of properties associated with Moringa olifera. And what we were interested in a specific medicinal property, that is anti-diabetic property. And now if you see the literature, there are a number of studies which have reported for Moringa olifera, but for Moringa congrensis, there are rarely reports come out. Now, this is one paper from Tamil Nadu where they have done in vivo studies for anti-diabetic properties in Moringa congrensis. Yeah. So as Nasir already explained about this transcriptome of Moringa olifera work, and this produced a number of paired and reads that around 271 million paired and reads from 5 different tissues. And we sequenced Moringa congrensis. We collected the plant from Indian Institute of Horticulture Research, Bangalore, and we collected the similar tissues, flower, leaf, seed, root, and stem, and we sequenced them in 3 biological replicates. And we generated around 705 million reads, and that we assembled. And we, since we want to compare with Moringa olifera, we assembled the Moringa olifera again along with the Moringa congrensis. Now we use previously reported genome from Moringa olifera as a reference to guide the assembly. And we quantified the transcripts in different tissues. Following to that, we have done some downstream analysis where we see the statistics here where we identify Moringa congrensis. We identified something like 42,000 unique genes and Moringa olifera around 32,000 unique genes. And you can see the BUSPO where we assess the completeness of the transcriptome where almost 90% you can see completeness for both of them. Yeah, so following to that, we went on to functional annotation where we could identify homologs for Moringa congrensis. 94% of sequences from Moringa congrensis, we could identify homologs in other plant kingdom species. Even we looked at the functional domains in PFAM database where we could identify like 77% of the sequences from Moringa congrensis and Moringa olifera sequences could identify domains. Yeah, so if we look deeply into those hits, what we can see here, most of the hits are from Theobroma cacao, where this species is evolutionary closely related to these 2 species, Moringa congrensis and Moringa olifera. And these are some of the top homologous hits. Most of them are kinases, and in the top functional domains, most of them are repeat domains. And we looked at the GOTEM functions for both the transcriptome, and we compared them, and we can see most of them are involved in binding, catalytic activity, so on. Yeah, so following to that, we looked at the gene family analysis, and here what is interesting, we looked at 2 species which are closely related to Moringa species, which is the Theobroma cacao, as we saw in the homology analysis. Also very closely related one is Carica papaya, and we selected 2 model species, Arabidopsis thaliana and Oryza sativa. So what we could infer from this, you can see Moringa olifera mostly close to Carica papaya, whereas Moringa congenensis you can see close to Pure Brahma cacao in terms of the cluster identified among them. So following to that, we moved on to the antidiabetic properties of these plants, where we selected 3 metabolites, secondary metabolites. One is quercetin, benzylamine, and chlorogenic acids. So there are reports these metabolites are involved in antidiabetic properties of the plant. Even in Moringa oleifera leaves, there are some review papers. These 3 compounds is involved in antidiabetic properties. And in quercetin, there are 7 enzymes involved. Benzylamine biosynthesis, there is only 1 enzyme. In chlorogenic acid, there are 3 major enzymes involved for the biosynthesis. Now, I'm not talking about the protocol as Nasir and Advait has previously talked about that protocol. I just want to take you through one example, that is 2 important enzymes from chlorogenic acid, which is highly similar by sequence identity. Now, using the protocol, we could identify the functionally important residue and the differences between these 2 genes. And when we did the phylogeny, you can see a clear a clear clustering between HCT and HQT, where you can find true hits based on our protocol. And hence, like this, we looked at all other enzymes from this 3 biosynthesis pathway, and we looked at the expression or the abundance of these transcripts in different tissues. So here, what you can see in Moringa congenensis and Moringa oleifera, the last enzyme or the key enzyme for producing the metabolites for each biosynthesis pathway, they all are almost expressed in leaf tissue. So that was interesting. There are reports that leaf is the most important tissue for antidiabetic properties already. Okay, so we validated this in whatever expression we got from transcriptome using qRT-PCR analysis for each of this enzyme. And following to that, we quantified these metabolites in the crude leaf extract. So hence, since the expression was mostly in the leaf tissue, we only selected the leaf tissue for this quantification. And we used methanol water extraction, and we did HPLC and LCMS profiling. And then now this quantification, we could find quercetin like 30 times higher quercetin content in Moringa congenensis compared to Moringa oleifera. And chlorogenic acid also we could find little higher content in Moringa congenensis. And for benzylamine, almost similar amount what we inferred. And we looked at the other metabolites coming up in the tissue using LCMS. Okay, now we wanted to see what are the antidiabetic activity having this crude leaf extract. Since we now we confirmed already like these compounds are present and in what amount this is present in the crude leaf extract, and we performed few in vitro assay studies such as alpha glucosidase inhibition study, alpha amylase enzyme inhibition study. And what you can see here, alpha glucosidase crude leaf extract mostly shown the inhibition. And we can see in lower concentration, Moringa congenensis showed better inhibition compared to Moringa oleifera. And here, Moringa alpha amylase, we could not find much inhibition. We also included benzylamine, which is an important metabolite for these plants because this compound is originally purified from Moringa plants, hence this also called moringinine. And we saw a higher inhibition for both alpha amylase and alpha glucosidase for benzylamine. And we also done DP4 enzyme inhibition analysis where we saw a clear inhibition for both Moringa oleifera and Moringa congenensis. And also we looked at benzylamine, and for DP4 also it started showing the inhibition. Now we from these in vitro studies, benzylamine showed in clear inhibition for DP4, alpha amylase, and alpha glucosidase. And we wanted to pursue more about this benzylamine. And we looked at the cytotoxicity of this benzylamine using MTT assay, and we seeded the cell and we treated with benzylamine. And after incubation, we checked the absorbance. And what you can see here, around like 4.5 millimolar concentration, it started showing less toxicity, but at the higher concentration, it started showing toxicity, and we used Hg2 CACO2 cells here, and now we added a little combustion of Moringa oleifera just to see how the toxicity is reducing. Now, you can see in the higher concentration the toxicity reducing drastically when we add a little bit of crude extract. Okay, so this is the work we have done for the Moringa congruencies plan, and we have compared with the previous Moringa drumstick tree, and we identified leaf tissue as an important tissue for antidiabetic activity, and we have done some in vitro study to prove that activity, and we published this paper last year. Yeah, so this work was supervised by Professor Sowdhamini ma'am and Dr. C.N. Vishnu Prasad from TDU. And the mass spec and other plant extraction work was, we got help from Dr. Radhika Venkatesan, and Dr. Sajeevan was helped in QRT PCR and RNA extraction. And the assay studies was mostly got help from Sanya Kausar from TDU. Okay, thank you.
Thank you, Shafiq, for the wonderful talk. So after hearing so many benefits of Moringa, we hope that Popeye the Sailor Man will start eating Moringa in place of spinach. Never mind. So moving on, next we will be in this today's talk, we'll be hearing about another beautiful plant which is called Sankapushpi. And this plant again has several medicinal benefits, which includes anti-convulsant properties, antidepressant properties, and also it can be used as a sedative agents. And most of these or some of these properties can be explained because of the presence of a interesting peptide called cyclotides. So in this work, the transcriptome of Sankapushpi was mined for such peptides called the cyclotides. And these cyclotides again impart several benefits, which can be pharmacological or agricultural point of view. And further, it has been shown that these cyclotides can protect against certain neurodegenerative disorders like Alzheimer's. This work was majorly done by Dr. Neha, but due to her unavailability, I would request ma'am to explain this work in detail. Over to you, ma'am.
Thank you, Vikas. I will start off with the presentation very soon, but since this is the medicinal plants part 2, there have been some queries following the part 1 as to how the lab and the team had selected or shortlisted plants that would be of use and could be quite attractive targets for future studies. And I would like to share the kind of ideas that went through our mind. First of all, we chose plants that are of Indian origin, and they would be multi-purpose in the sense they would cater to curing multiple diseases as documented in Ayurvedic texts and in the literature. And also it should be a fairly popular one in the society so that people can connect with such plants. And also we selected these plants such that your genome or transcriptome data are not yet available. And we had not considered other factors such as whether there is a nearby plant whose genome or transcriptome is available, because we were kind of confident that we could do de novo assembly if other factors come in useful. And we had also not been able to consider genome size because that requires either such information in the literature, or we have to carry out our own experiments. And we had also not considered the ploidy of the plant. So that was the little bit I wanted to share about how we had shortlisted the plants. Yeah, so about the Sankapushpi, I must say that this was not in our list of shortlisted plants. We had not, recognized this, but this plant was brought to our attention by means of our collaboration with both Professor Balaram and Dr. Radhika Venkatesan. And in fact, Professor Balaram was a major driver for this project. And of course, our lab was very attracted to take this up because it kind of meets all our criteria that I had just mentioned. Plus, the plant is also having these sort of cyclotides, as Vikas mentioned, that are not only cyclic polypeptides, as the name suggests, but also they are endowed with as much as 3 disulfide bonds. And you can imagine together the amount of stability it can impart to such a small peptide. And indeed, these cyclotides, as synthesized by the plants, are ultra stable, and so stable that if you are interested in extracting the cyclotide, all you have to do is to boil the plant extract, and you should be able to isolate cyclotides. As mentioned, the plant, this Sankapushpi has been used in over and over again for many years, and the tribal populations, for instance, had used it for treating people, ladies who are going through a childbirth, because it can reduce the pain that is associated with that event. And also it has a number of other properties that have already been mentioned. Okay, so these are the kind of scenario in which we selected the Sankapushpi. But let's look at the cyclotide itself. And these polypeptides are not only observed in Sankapushpi, but few other plants, and these are shown here. And you can see them in Fabaceae, where Sankapushpi is one of them, and it's also observed in Rubiaceae, Violaceae, Cucurbitaceae, Solanaceae, and the plants like coffee. However, what is equally interesting about this particular polypeptide cyclotide is that, unlike the secondary metabolites that are associated with medicinal value in the other medicinal plants, this happens to be a protein itself. Although we finally see a small part of the polypeptide, which is in cyclic and endowed with disulfides, the original protein that codes is actually the pre-pro form. This has some remote resemblance, and some of us might recall chronotoxins that also have similar properties. But that aside, you can see on the left-hand side the domain architecture. And what we are looking at the end of the post-translational event is the cyclotide domain shown in yellow color. So I just wanted to bring home the point that this arrangement of what all goes into the pre-pro form could be somewhat different when you look at the different plant species. They are not universally conserved, but the cyclotide domain itself is structurally similar. All right, and we can also see that much amount of sequence and structural information are already available for the cyclotides, and indeed structures are available in the protein data bank, and it has been classified into the type 1, which is a Mobius, and the type 2, which is bracelet. And Mobius is somewhat a kind of a structure where you have a one-dimensional part, and the other rest of the cyclic moiety is considered to be twisted up. And hence the name Mobius has come to this conformation of cyclotides that adopt this structure. Whereas you can have a variation which is called as a bracelet, and between them you can see some differences at the level of secondary structures, small helices coming and going, but also at the loop 2, one of them contains a cis-proline. So therefore, there are key structural variations between the two classes of cyclotides. But what is rather nice to appreciate is that the disulfide bond connectivity itself, which is shown on the top right, is fairly well conserved. And in between the cysteine residues that form the disulfide, there is substantial amount of sequence variation that can suggest that on the one hand, you have these key motifs, but on the other hand, you can have a lot of sequence diversity. That means you have to be rather cautious when we start to look for cyclotides in new plant genomes. All right. So that's about the introduction. And then what had been explained very nicely by Nasir and later by Shafi on the different Moringa varieties, here also we had gone through the same procedure of collecting, purifying RNA and then collecting, in this case, we had as many as 282 million reads, and these were all assembled using a series of softwares that got trained up for this plant. And then we were left with the statistics as shown on the right-hand side. The N50 values were more than 2000, which is quite good. And then it was sufficient to carry out the investigation of the cyclotides in Sankapushpi. And we also, like in the Moringa, concentrated on a number of tissues, namely flower, leaf, pod, and stem. And then Neha had carried out the rest of the analysis. So the next 4 slides, I'm going to concentrate on what we learned about cyclotides in Sankapushpi. And after that, I'm going to spend some time, say 3 slides, looking at the other enzymes that are involved in this pathway of the production of the cyclotides by these plants. All right. What we have about the cyclotides back to remember is that it contains multiple domains in the pre-pro form. So, however, when we are looking at the transcript, this is prior to the post-translational modifications, right? So we can anticipate the rest of the domains are also observed in the transcripts. So we use them to recognize the cyclotides, and the cyclotide domain itself is shown in the blue color, and you have some parts or flanking, some domains flanking either side, right? And then you can also see that when we look at the alignment, the cysteine positions are conserved. That means the 6 cysteines are there that are responsible for those 3 disulfides, which we had appreciated when we looked at the structures and so on. And we also managed to identify in the Sankapushpi transcriptome as many as 71 cyclotides, which is rather nice because in parallel to what we had seen for the phonotoxins, for the ponus snails can usually produce a battery of some closely related sequences of phonotoxins. In these plants also, they are known to produce a number of cyclotides, and of course, these are spread around in different tissues, and here as well. And out of the 71, 26 of them turned out to be novel, and this was sufficiently supported by mass specs, which I will come to very soon. But what we can see is that we were able to recognize as many as 71 of them. And how do they look like in terms of their tissue distribution? And this is possible because we do have the transcript from different tissues. And what we found, to our utter surprise, was the hierarchical clustering of the cyclotides, of the 71 cyclotides in this plant, on the basis of their sequence diversity, neatly seems to correspond to the tissue localization. You can see that the one cluster, and on top of that is overlaid the heat map of the TPM values. And you can see that each cluster seemed to have already recognized which tissue they should be. And for instance, we have now zoomed up one part which corresponds to the pod, and that neatly corresponds to one of the sequence clusters. And of course, it has both bracelet and Mobius. All right, and the next slide would show that. But before we go to the kind of clusters, the sequence clusters in some detail, I wanted to say that this particular project is multifaceted because it uses multiple techniques, not only transcriptome data, but since the other collaborators were experts in other areas, we could use mass spectrometry. And this was done by Neha. She collected the samples now from the different plant tissues, and then submitted it to mass spectrometry, both LCMS and MSMS. And here you can see that the LCMS itself vividly shows the huge number of cyclotides that can be witnessed, say in any one of the tissues. And then the question really is, can we use MSMS to decipher the sequence? And this kind of corroborated very well with the transcript data, because this is going to give information as to which residue is next to which residue. And clearly we could augment what we had seen from the TPM data or of the transcripts to then go into much more finer detail and ensure that the sequence we see from the transcript data corresponds from the mass spectrometry data. And these are shown here. And somehow our attention also got attracted to not only looking at the cysteine residues, but some of the proline residues that seems to also have some specific positions and patterns were emerging. So that aside, back to those sequence clusters, this particular mass spectrometry work was published by the 3 other people in the team, which are Neha, Professor Balaram, and Dr. Radhika Venkatesan. And this was published in Asian Journal. In fact, this came also as a cover picture, and it was very rewarding. But back, how did it help us with these sequence clusters? We can see that not only there is tissue specific kind of preference, there are 3 main clusters. When we try to use sequence diversity as a dissimilarity as a measure, and cluster A and B, for instance, are of the bracelet type, those 2 structural types that we had discussed, whereas the cluster C was of the Mobius type. And that particular aspect came quite nicely, and that's all about the cyclotides in Sankapushpi. But we extended this question with the availability of very high quality transcriptome data to ask how the enzymes that are associated in the synthesis of the cyclotide itself, because some enzyme is going to be involved in the cyclization, some enzymes are going to help the disulfides to form properly. So can we follow them? So with this question in mind, and knowing that these enzymes, such as here, what is shown is the protein disulfide isomerases, would be in different parts of the cell. As the cell is synthesizing these beautiful cyclotides, we went after the sequences, starting from queries, as already very nicely explained in the CAPS protocol, the pipeline by Advait, and recently also by Nasir and as well as by Shafi. So using these sort of queries, we looked for the protein disulfide isomerases, and then we found them as many as 15 different genes, transcripts rather, in the transcriptome of Sankapushpi. And as you can see on the right-hand side, some of them which have been shown in ellipse correspond to only the Sankapushpi, whereas we have included some of the annotated and trusted protein disulfide isomerases from other plant species. So whereas some of the cyclotide identified PDIs, protein disulfide isomerases, co-clustered beautifully with the other with those of the other plant species, you can also see some clusters which have been shown in ellipse, which are containing only PDIs from cyclotide, from Sankapushpi, suggesting that these are plant specific. And we can understand this because, after all, if the plant is producing a huge variety and numbers of cyclotides at different tissues, perhaps it requires multiple PDIs to help in this process. And definitely 15 different PDIs have been put to use for this very specific purpose. And we can also appreciate that the domain architectures of those different PDIs are also somewhat different at the transcript level as to what are the coexisting domains and so on. So this is the learning we get about PDIs. And we also closely followed 3 more enzymes. And the next slide shows that of the enzyme that is most important because it catalyzes the cyclotide cyclization. And this is called as an aminopeptidase. And in the biotech industry, as we understand, as we started explaining, Neha started explaining her work, biotech industry people are very much looking for such cyclization enzymes. And to our luck, we found that not 1, but as many as 7 different amino acid asparagine endopeptidases could be observed in the transcriptome of Sankapushpi. And these are again marked up, and you can again see that some of them occur as Sankapushpi specific clusters, suggesting that multiple ATEPs are required in this particular plant to ensure that there is sufficient amount of help or catalysis of this last step of the cyclotide formation itself. And in addition, we were also looking at a couple of other enzymes such as ERO oxidase and ERO oxidoreductin, and definitely the cis trans isomerases. And we found just 1 evidence of ER oxidoreductin in this particular plant, but we also could evidence as many as 70 different cis-trans isomerases, suggesting that this particular enzymes, PPIAS, peptidyl-prolyl cis-trans isomerases, might have been recruited in multiple other pathways. Definitely, they are not all engaged in the formation of the cyclotide, but they must be required for many other situations. And interestingly, their domain architectures are also different from each other amongst the 70 examples. All right, so this was about the transcriptome work and the way we had mined and looked for enzymes of interest, starting from the special metabolite, which is the cyclotide, and the enzymes that are enabling the synthesis of the cyclotides, such as we had talked about oxidoreductases, proline disulfide isomerases, the cyclization enzyme, endopeptidase, and the oxidoreductant, as well as the peptidyl-prolyl cis-trans isomerases. Okay, so this was mined using transcriptome assembly data, but then Neha went on to do a few other aspects as a part of her PhD. First of all, given that we now have as many as 71 different cyclotides, and the structures must not have been available for all of them, right? Because there are a few handful of structures in the protein data bank, but we wanted to address this from first principles. The fact that they are all rich in disulfide bonds was exploited, and we Neha used a technique, an algorithm called RANMOD. And RANMOD was written by me in my PhD days at the latter part of my PhD, when I had become sufficiently independent to come up with such ideas. And essentially, this method exploits the property that a polypeptide that is rich in disulfide bonds have not many secondary structures. Here, globular structure may be driven by the presence of multiple disulfides. Plus, each of the residues, however, although they may not form secondary structures, they have still got a constraint to be in allowed regions of the Ramachandran map. So the fact that we had a very reliable procedure called MODIP, which will permit us to recognize sites where disulfides can be incorporated in a strainless manner using side chain stereochemistry, formed the heart of this RANMOD algorithm, wherein we begin by knowing definitely what is the sequence, primary structure of the polypeptide, such as cyclotide in this case. And then we would also like to know whether they are the glycine type, the proline type, or the non-glycine, non-proline type, and assign appropriate backbone conformations. And we would also like to know the disulfide bond connectivity. With these two sort of data, the algorithm called RANMOD, which was written by me years ago, would assign start by random numbers. It generates random numbers, and they get transcribed into phi, psi, and omega values. Depending on the amino acid type, it will assign it to appropriate phi, psi values, which are random but stereochemically allowed. For instance, if it is a proline residue, it will fall in this thin allowed region that corresponds to prolyl allowed regions of the Ramachandran map. But if it's non-glycine, non-proline, it has a few more variety, and if it's glycine, it has a lot more freedom of space. So after a while, this was going nicely because these are small peptides, and that means you do not have to create too many random backbone conformations. And somewhere, as we start to sample conformation after another, at least a handful of them would click. In the sense, the disulfide bonds of interest are indeed possible, as guaranteed by MODIP algorithm, in the sort of positions that the random conformation has just been assigned. And further, they can be recognized as whether it's high quality disulfide by means of different grades. On this sort of an approach and validating the RANMOD, Neha chose 3 different peptides. One is the cycloviolacin, which is actually a cyclotide, but from some other plant, as we had earlier seen. And she also applied it on 2 other peptides, which are small and disulfide rich, such as phonotoxin which we had been talking about a lot, and another polypeptide, which is also small and disulfide rich. This is the trypsin inhibitor. And in each case, we were able to recognize and come up with a grandma generated model that closely had resembled the structure, either derived by NMR or crystallography. So this encouraged us to look into the structural modeling of cyclotides. All right, so this was the modeling sort of parts. But I still have two more angles to present of the Sankapushpi, and all the work was pursued by Neha. And for instance, knowing that the Sankapushpi is endowed with a huge number of medicinal properties, we decided to focus on one of them, which was mentioned by Vikas in the introduction, which is its capacity to revert people with neurodegenerative diseases. For instance, it is implicated in relieving diseases such as multiple sclerosis, and it has also been documented to be a potential inhibitor for amyloid deposits and tau aggregates. And this has been documented in different publications, as you can see underneath. And since this is the case, Neha then went on to carry out some behavioral assays. And for this, she had in her hand quite good extracts from Sankapushpi. So the question that she asked was very simple. If we were to follow through the sort of assays that actually some people in TDU, such as Ashwini Godbole, had been using to compare transgenic worms, which are which have been purposely paralyzed. And what you see on top of in the video is a normal C. elegans worm, which is moving around reasonably fast. But on the other hand, this transgenic worm is which has been endowed with a lot of amyloid aggregates, is hardly able to move. So that means it has gone through the paralysis, clinically speaking, as it were. And the assay would then be to measure the extent of paralysis when this sort of amyloid deposition has been induced in the transgenic worm after increasing the temperature, say from 16 degrees to 23 degrees. And then we would start to examine the extent of paralysis at the end of 96 or 112 hours. All right. And what the question really Neha was asking is, in the presence of the cyclotide extracts, how are these worms behaving? And for this, she compared the worms at when there is no paralysis, that namely the wild type, which is shown as a constant, a baseline, where 100% of the worms are not expected to go through paralysis, right? Because there is no aggregate. On the other hand, the transgenic worm has been induced and imparted a lot of these deposits, aggregates, and that is what is shown in the negative control, which is the one that is dipping down. And it dips down over time because more and more deposits are there, and the percentage of the worms that are getting paralyzed is not getting paralyzed, is becoming lesser and lesser, right? So that is the wild type on top, and this is the negative control where there is a huge decline. But what we want to see is compared with the positive control, which is a sort of well-accepted drug for relieving paralysis. And that is shown in this kind of open gray circle, that plot. And you can see that somewhere around 50% of the worms, however, are still gone through paralysis. And what happens to the Sankapushpi extract at different concentrations as injected by Neha to the worms, to the transgenic worms. At a lower concentration of 20 micrograms, it is not catching up as much as the positive control. But at 80 micrograms, at 200 micrograms, it is easily surpassing the positive control, suggesting that Sankapushpi indeed can have a relief against paralysis as after using C. elegans as the model organism. All right. So this was the part, and she also ensured that the amyloid deposits are reducing over time and in the presence of the cyclotide extracts. And this is what is shown in the controls, where there is sufficient amount of deposits you can see, and even in the positive control there is some deposit. In the thioflavin staining, what Neha finds is in the presence, after injecting the Sankapushpi extract, the amount of deposits have significantly reduced. You can see them as small nodules, but not spread around the entire worm. So these are kind of two independent ways by which she has used behavioral assays and imaging to show that Sankapushpi extract can help with the reduction of amyloid deposits. And how do they do that in a mechanical form, in a structural form? And for this, we decided to do some molecular dynamics. And that is what I'm going to show. All right, so this part of the work of Neha had been published in Journal of Medicinal Chemistry. And again, it has come in the cover picture. That's very good for us and Neha especially. So the molecular dynamics simulation had one purpose. So what we would do is to go into the protein data bank, collect as many amyloid structures as possible, and you can see them all on top at different levels of aggregation. We do docking of one of the cyclotides of Sankapushpi against each of these molecules, amyloid deposits, and select the best pose of pores, and then subject them to molecular dynamics and analyze the different amyloid aggregates with and without the presence of the bound cyclotides. And to our encouragement, we found, first of all, the cyclotide binds and it remains bound at the end of the simulations. And in fact, there are strong hydrogen bond and hydrophobic contacts between the cyclotide, which is shown in mild blue color, cyan color, and the amyloid deposit is shown in green color. But what happens, interestingly, at the end of the simulation is the aggregate part. This is to show that the aggregate is still stably binding to the cyclotide, but what happens to the aggregate itself? It seems to have lost its intention to form a strong interstrand hydrogen bonding, and it's now in the presence of the cyclotide, it seems to have completely lost its capacity to form the beta sheet, and several hydrogen bonds are indeed getting lost. And what is shown on the top panel is without the cyclotide, or shown at the look panel is with the cyclotides. I feel that this is very, very encouraging. And overall, we are able to take forward the work on Sankapushpi through different directions, namely transcriptome, and then doing some amount of three-dimensional modeling, doing behavioral assays and imaging, and also supporting with molecular dynamics data. And this part of the work had just been published in Frontiers in Molecular Biosciences, where Neha had done the primary work. All right, so I would stop by acknowledging the key people who are involved, Neha, who had carried out all the work, and in collaboration with Dr. Radhika Venkatesan, who had guided Neha on the plant extraction, and also on the mass spectrometry work was guided by Prof. Balaram. And I do think the way Neha could move from one area to the other was simply possible because of the motivation provided by the co-guides. With this, I would stop. Thank you.
Thank you, ma'am, for such nice explanation for the work. So these were the three topics for today's talk. Next, I would request Shafi to put up the relevant publications for these three topics. Over to Shafi.
Hello again. So these are the relevant publications from today's talk. And the first part, Moringa olifera, Nasi talked about this work, and we have two papers in their work. Actually, three papers. One is the method. Earlier we talked about them. And here the first one is the transcriptome profiling of Moringa olifera, and the data was published in a companion article. And second one, myself presented the Moringa competencies work. So the third session, ma'am talked about some Sankapushpi transcriptome work along with the other disulfide rich peptide work and other mass spectrometric analysis and also molecular dynamics simulations. Okay, so these are the relevant publications. And next week in CAPS talk, we have the last talk in this theme, medicinal plant and collaborations. We have titled the talk as beware, we can read your genomes. We will listen about one invasive plant and also bacterial and viral genomes. Please stay tuned.
Thank you, Shafi. So these were the three topics for today's talk. And so these were three speakers for today's topic, and their current affiliations are as follows. So Dr. Nasir is currently the lead bioinformatician at ZS Associates. Shafi is a postdoctoral research associate in CAPS lab itself. And Neha is postdoctoral research associate in Carol Robinson Group at the University of Oxford. So with this, we conclude today's session. I request the speakers to stay on. Thank you, and we'll meet next week, same time. Thank you.